Now, hi guys. So earlier we have seen that the basic difference between animal and plant cells. There are loads and loads of differences, but in this particular case, we have studied four major differences which are much more important than other differences, right? So now let's move on. How should we draw a particular eukaryotic animal cell, right? So in case of a particular eukaryotic animal cell, we have to consider and the different kinds of diagrams, and then we will go for the topic one by presenting the different cell organelles, right? So let's move. Let's see how we can draw it easily. So it's a freehand structure and very easy to draw. Okay, so let's move on to our next structure, that is the eukaryotic cell, animal cell. I'm drawing an animal cell right now. So first in here we'll get some finger-like projection, and then from here we'll get a structure. Now what cell invagination in there, then another cell invagination. It looks quite good, maybe too many cell invagination, right? So this is mainly the structure of an animal cell, and these parts will form the Bird's curves or cell invagination that will help us to understand the cell membrane gaps and joints between them. So for this topic we'll concentrate on this one right now. So here we will have a nucleus, a nucleolus in here, then some chromatins. I'm drawing just a freehand structure, remember. Okay, then we will have some mitochondria in here. It will float around the cell everywhere, just have some elliptical shape in here, and then give it a spiral. This will define the Christie of the mitochondria, right? Then in certain eukaryotic cells like sperm cells, we can have a tail-like projection known as this tail-like projection in here. This is known as flagellum. Actually it turns in this way and helps the cells to move forward, and in certain cases we can find certain cilia in these cells, right? Cilia, we will find these cilia in this kind of cells, but in case of our lungs respiratory cells, right, the cells that constitute the alveoli or the mucosal membrane, we will find that these kind of cells where we can have certain cuboidal ciliated columnar cells, right? Ciliated columnar cells, so they will be on this side, okay, not on the full side. We can draw it in a particular single side. So we can move this one, right? And then afterwards what we will go, we can add certain cilia in here, okay? So there will be certain cilia, right? Now what we'll have, we will have some lysosomes in here. That's a freehand structure, remember I'm telling you, and certain gastric material, that's the cell debris we can see, right? Now you have to remember that with the nuclear membrane, which is a bilayer membrane, so just we will give it a knot of a second line, right? And then what we will draw in here, we will give the structure of endoplasmic reticulum. Lots and lots of people may make mistake about these structures, right? So endoplasmic reticulum is associated with the nuclear. Outside the nucleus you will find the endoplasmic reticulum and it's quite easy to draw in here. Like this one, and from this side you can have this one. Okay, we will not make it that much large because we need place for the Golgi body also. Okay, so what we'll do, we'll just delete some lines from here, and then we will add something like this. Add these lines in this way. Okay. Look at that. It's quite easy to draw. This structure is quite easy to draw. Right. Then from here we will extend one part of endoplasmic reticulum up to the cell membrane, right? This is our cell membrane, so we will move up to the cell membrane in this way. Now remember that the nucleolus generates some structures that are known as ribosomes, which helps in the synthesis of protein. So we will draw some ribosomes in here, and that has to be closer to the close to the nucleus, and then rest of the part we will leave it because that is our smooth endoplasmic reticulum. Okay, now then we will have a structure which will contain certain microtubule structure and astral rays. So this is quite a funny structure, and this structure is known as centrosome. In here we will be drawing certain smaller structures that helps in the formation of enzyme and packing of protein, and that will be known as the Golgi body, right? So in here we will add some granules